right, good morning, everybody. We're going to get started in a minute. Or if you're real quiet, we'll just get started now. Good morning. My name is Toby Shepard. Welcome to the Chautauqua Lake Conference. So happy you're here. It's a beautiful morning. I was thinking about a couple of years ago when we were here last. It was like 55 degrees and raining, so we'll take it. It's a nice and sunny day. I've got some logistics to go over in a second, but right now I've had the pleasure of introducing Chautauqua's president, Dr. Michael Hill. He's our 18th president, and just a bit of background. 150 years ago, Chautauqua's founders imagined a place where citizens could make purposeful use of leisure time through experiences in education, religion, recreation, and the arts. This summer, Chautauqua celebrates its 150th anniversary of that incredible vision that is still relevant today. Michael's leading the institution through a strategic plan, 150 Forward, and that plan's at its midpoint. But from the beginning, creating science-based solutions to the conservation of Chautauqua Lake has been among the institution's top priorities. That prioritization has garnered important partnerships, including that which we enjoy with the Jefferson Project. It's also brought significant resources to Chautauqua Lake, including Governor Hochul and the New York State Legislature's recent allocation of $1 million to support the work of the Jefferson Project here at Chautauqua Lake. Diversifying the institution's revenue streams has also been a priority of this plan, and as a result, Michael is at the helm of the largest capital campaign in the institution's history, which has raised $114 million to date out of a total goal of 150, which is pretty awesome. Prior to his appointment at Chautauqua, Dr. Hill served as the president and chief executive officer of Youth for Understanding USA and in senior external relations roles for the United Cerebral Palsy, Washington National Cathedral, and the Washington Ballet. Michael, we're so happy that you're here with us this morning.
So Toby may be happy with the weather, but for those of us wearing suit jackets, I'll take 50. My apologies for my tardiness. When you get programmed for about 17 events in a day, sometimes your calendar has the wrong location. I was ready to greet you all at the hotel. So, but good to see you. And thanks, Toby. It's good to see you back here as well. And thank you all for making time this morning on what really is the start of our season. Hopefully you'll be with us for Martina McBride this evening if you can. Toby mentioned our founders, and we've been thinking a lot about that in our 150th birthday. They were two very different people with different backgrounds. Lewis Miller was a businessman from Akron, Ohio, and his partner was John Hell Vinson, who was a bishop in the Methodist tradition from the South. These men really didn't have a lot in common, and as I understand it, they had some really famous arguments throughout their time here at Chautauqua. But these two men with different backgrounds and different ways of looking at the world had one thing in common: their belief that education, including religious education, was essential for productive citizenry. And on this, Miller and Vincent violently agreed. And it occurs to me this morning that their partnership is a lot like the partnership that we have in this region. We often don't agree on the next best step when it comes to lake conservation. Goodness knows we don't agree and battle often on the use of herbicides. And we don't even agree sometimes on what problems we're trying to solve for. But we share one important thing in common, like Vincent and Miller did. Everyone here cares deeply about Chautauqua Lake and wants it to thrive. We might even agree that this lake and what we're doing together to save it can stand as a model for others to follow. We're certainly not the only region struggling to save a crucial natural resource, but this thing we share, this commitment to making this lake better for generations to come, is our reason for being here this morning. As it was for Chautauqua's founders, a common purpose can be a powerful thing for us. A common purpose can create something that never before existed, but that the world desperately needs. A common purpose can withstand a lot of disagreements if we all just commit to continually returning to the table, regardless of whether we won or lost the most recent battle. A common purpose can sustain a community and maybe even a lake for an eternity. And so we're here today to nurture our common purpose by sharing knowledge and perspectives and by striving to listen deeply to understand. I look forward to all the learning that we'll do together today and what that will allow us to create for tomorrow. And I want to thank you for doing this and thank you for taking this opportunity. I do want to just take one last moment of personal privilege. Toby mentioned the state of New York, and we have been served mightily in our region by someone for a very long time, who is our Assemblyman, Andy Goodell, who came in and picked on me before I got up here. Andy's retiring this year. We will miss him tremendously. He has been a ferocious advocate for this institution in this region, and I think it's fitting that we start our day by thanking Andy for all he's done. Have a great morning.
Okay, thanks very much, Michael. A few logistics. Yeah, in 1 second. Yeah, you can do it. A few logistics. If you're not familiar, just make sure this bathroom's back here. And you probably saw the coffee and snacks. Make sure you're getting lots of coffee and snacks today. On the Q&A, so we'll have several presentations today, and in Chautauqua style, of course, there's going to be question and answer. So if you want to use, there's cards on that table up there and some pens. There's also cards in the back. If you can write your question on a card, and we'll collect them. I'll try to do my best to sort of categorize questions so that we can get the most bang for our buck from our presenters today. Okay, and now that logistics is out of the way, let's get to our presenters. Our first presenter is Dr. Courtney Wigdell-Perry from SUNY Fredonia. She is an associate professor of biology there, and she earned her PhD in ecology and environmental sciences from the University of Maine. As an aquatic ecologist, Dr. Wigdell-Perry's research program centers around understanding how lakes respond to environmental change. Sounds pretty good. I'm glad she's here. She works on local lakes in Western New York, as well as other systems around the world, primarily focusing on algae communities and water quality issues. Today, Dr. Wigdell-Perry is going to give us an update on SUNY Fredonia's research program and their plans for 2024.
Okay, great. Time going. All right. Thank you so much for having me here today. It's great to be here. I look forward to this every year, so thank you especially to our hosts for organizing this and having us all back again. As scientists, we don't always have a chance to share our work back to the community that it impacts so much, so we really appreciate getting to do that, and thank you all for being here today. And you get to enjoy some AC, so that's always really good. I know we've been melting all week up in Fredonia. So as Toby mentioned, my research program at SUNY Fredonia is focused around this question of how lakes respond to environmental change. And you'll notice throughout the talk, I've tried to bold the questions to make it really clear what we're trying to tackle with all the different things I'll be sharing today. A lot of times I sort of think about, I'm mostly interested in algae, so why am I studying all these other aspects, right? If you want to know what's going on with algae and cyanobacteria in a lake, you need to think about all of the other things they're interacting with. The abiotic or non-living environmental conditions and the biotic interactions all shape what those communities are doing, and ultimately what we see in terms of blooms and water quality issues that we're so concerned about. We're a primarily undergraduate institution, so I have to give a big shout out to my students. So we have mostly undergraduates with a small master's program. And so the work that comes out of my lab is done by students, driven by students, and they bring these questions to light, and it's really amazing to watch them grow as scientists. And it's not only are we doing cool science to learn about the lake, but we're also providing really great opportunities for students to grow. So you can imagine with all these different students rolling through the lab that we have lots of questions and lots of projects happening at any given moment. So for today, I'm just going to talk about a couple pieces, but if there's something I don't cover, certainly find me later, and I'd be happy to talk about some of the other projects we have going. We don't need to sit here for a week and hear about all the stuff that we're up to. So I've just picked 3 vignettes to tell you about, 2 from my graduate students who just finished up their master's theses in May, and then another at the end looking at sort of the year-round perspective and trying to push us beyond just the summer season. So I want to come back to this theme that we've hit fairly often in the last couple of years, looking at vertical temperature patterns. And so we really want to understand what's happening with temperature in different parts of the lake, and primarily around this process called stratification. So in deeper systems, just as a quick refresher, if you're not familiar, we have during the summer months this sort of warm layer that overlays this cool deep layer. And it happens because of density differences of water at different temperatures. And what we end up with is 2 different environments basically that develop in the surface versus the deep water. This matters for algae communities because as you have these different processes happening in these different layers, we have changes in nutrients and other kinds of water chemistry issues. Oxygenation, the physics and how things move is different from the surface to the deep. So we're seeing blooms at the surface, but that's not the whole story. We need to think deeper than that. We've been tackling this problem in our lab using high-frequency temperature loggers, small, simple, but very many of them, and very powerful en masse. This fleet of temperature loggers are deployed on subsurface temperature lines, subsurface sensor lines, and we sink the float at the top of the line about 5 to 6 feet below the surface of the water, so it's safe for boats to pass over, which means that we can put them wherever we want to, which is very helpful. Getting them back is the interesting part, but I can tell you about that later. So we have a concrete block at the bottom, and then these sensors are distributed every 0.5 to 1 meter, and they log at 5-minute intervals. So we have really increased our understanding of spatial and temporal scales by taking this approach of deploying this fleet of temperature sensors around the lake. We've also been able to deploy at the base of these lines some sensors on loan from Kevin Rose at the Jefferson Project and RPI so that we can get oxygen values at the bottom of the system in conjunction with the temperature, which is very cool. We've been tackling this since about 2019. Actually, I have some former students here, which is very exciting. So my student, then Mason, now Shaman, is here, and she started this whole story back in 2019, exploring data from the North Basin with these sensor lines. What was really fascinating is that we thought that the North Basin was relatively firmly stratified, but what Mary found is that that boundary between the warm layer and the cool layer is not static. It actually flexes and sloshes back and forth. And I mentioned that in past years, but if you haven't seen it, it's very cool. And that comes into play a little bit later. So I'm going to keep that in mind. We're going to come back to that in a moment. Another student that I have has continued to work from an undergrad to a master's degree, and Casey's here. So yay, Casey. We got all, like, Fredonia alums are amazing. They keep coming back and keeping me posted on what they're up to. It's great. And so he investigated in the South Basin, all the different locations in 2021. And then we started to say we need more information synchronously around the entire system. So we started pushing that out to the North Basin, the South Basin, and this middle section in between in Bemis. As we were looking at this, we realized that this middle portion, if somebody has a better name for it, please tell me, because I just call it Bemis Bay, but it doesn't have, like, the unit doesn't have like a good name, right? It's more than just Bemis Bay, but it's this interchange, this interface in the middle of the lake. And it's comparatively understudied when we look at what kinds of data sets we have for the North Basin and the South Basin, and with good reasons, right? You got 6,000 acres to the north and 6,000 acres to the south that are important. And this sort of sometimes gets overlooked as just a pass-through. But when you have a very different physical environment in the North Basin, a much deeper system that has all sorts of different processes happening, and that meets with the South Basin, what happens, right? The shallower, warmer system, we had lots of questions. And so this is what we were trying to get at this critical junction was, what does Bemis Bay do in terms of stratification? Is it an extension of the North Basin? Is it an extension of the South Basin? Or does it do its own thing? And what kind of factors are important in this process? So this was Casey's work for his master's thesis. Again, he just finished in May, so these things are hot off the presses from what we've been working on. Fortunately, we've known this for a while, that we don't have a lot of data from this area. So we've got some more recent data from the Jefferson Project's efforts to do with their sampling in this area throughout the whole lake, and also some very interesting hydrodynamic modeling, looking at the surface versus the deep currents in this region that they've talked about before. We started investigating this more in 22 and 23 with those sensor lines that I just mentioned in Bemis. So I want to show you just a quick temperature map. You'll see these heat maps and other sorts of cross sections of data through time. And so we have the surface zero on the vertical axis going down to the deepest portion of Bemis. We were anchored about 6 meters. And then we see this through time. I don't have the whole summer season. We do have shown here, but we do have those data from 22 and 23. But what you'll see here for this week in July is, again, that warm temperature overlay, but that periodic stratification event that occurs, right? What we're seeing in Bemis is that it's not behaving like the North Basin, right? It's not stably stratified. It does occur, so it's a little bit more like the South Basin where we have that intermittent stratification, but it's not synced up. It's not exactly the same as what we're seeing there. So it seems to have its own distinct pattern. And we wanted to know why. What's driving that? What are some of the factors that control this? So Casey took the bold and brave dive into modeling to look at predictive statistical modeling to try to understand when the lake occurs, what are the conditions that result in that stratification event. So we used multivariate adaptive regression spline, or MARS models, and it basically fits a model using all of the input data and gives us the simplest model possible that gives us the most explanatory power. We used 2 summers worth of data to put into it. So we built the model on 60% of the data and then tested it on the other 40%. And our inputs were external variables. All the weather data that Casey could find went in, so air temperature, humidity, air pressure, precipitation, all of those things. And then we actually also included that North Basin SAISH flex. We had a metric for that to try to understand what's happening as that is shifting at long point as it's coming around the bend. Could that be important in shaping the story as well? The models are assessed based on fit to our observed data. So we run the model, we predict, and then we test it to see how well it fits with what we saw in our data. I'm not doing a lot of deep dive here, kind of keeping it tight, but we did, Casey really, not me, not we, Casey, developed a successful model that if you look at our vertical axis, you see our predicted stratification index from the model compared to our observed data. And you see we do really well. We have a good predictive power. We do predict the events. We underestimate a little bit on the intensity on the high side, but overall the model works quite well. And what was really interesting is that it didn't until we included that North Basin connection factor, right? The model, we basically kind of doubled our predictive power by including that interbasin connection. What we think might be happening is that as that internal SAISH rocks, that mass of cold water sort of gets pushed into Bemis. We don't know for sure. That's what we suspect. It seems to make sense based on what we're seeing, but only under certain conditions. So we have a lot more to learn, but this tells us really, we can't look at any of these portions of the lake in a vacuum. We really have to see the whole story together. So while we talk about the North Basin versus the South Basin, we have to see that story and the connections in between. The next story I want to tell you is looking at road salts. This is a very different story, shifting more into the chemistry world. I mentioned this last year, gave you a bit of a preview. This is what we were planning. This was a project for my student, Cameron Finch, who also finished in May for his master's degree. The question that a lot of people ask is, what is going on with road salt at Chautauqua Lake? Because people at other lakes are also asking this question. The bottom line is we have not done a very thorough investigation yet. This is something we really have to learn more information about. So we started doing a dive into this, and we were looking at some of the data that we do have for chemistry within the lake. The conductivity, which is the sum of all of the ions, all of the salts, not specific to road salt, but sort of a grouping of all of that, right, affected by many things. Based on the 2022 CSLAT volunteer report, the analysis suggests that there is a significant positive trend and an increasing amount of conductivity for both basins. I'll hedge that by saying that the analysis changed in 23 with the report that came out not too long ago, which says there is no trend. So there's some work to be done in terms of figuring out what's really happening. If you look at the data, though, you can see that it goes up a little bit, comes down, goes up. Not a strong, dramatic trend. We're very much in the range for freshwater systems in terms of what it is. So we're not like in crisis mode, but that's good. We don't want to be in crisis mode and then start asking these questions. We know right? We use a lot of road salt to keep our roadways safe and make sure that people can travel safely in the winter around here. But now's a good time to start asking this question. And we were in particular interested in what is happening with salt tolerances or sensitivities in our zooplankton community. Most of what I do is algae, but zooplankton are the animal plankton that eat the algae. They're the next bump up in the food web. And they also serve as an important food source for larger organisms, so larger inverts and fish, and then for the really big fish. So zooplankton are an important part of that story as well. And also relatively understudied in our system. We started down this pathway because we had an opportunity to work with colleagues at the Global Lakes Ecological Observatory Network, GLEON. It's a global grassroots group of scientists and lake managers and government groups and lake organizations to really understand what's going on in our lake ecosystems. Dr. Shelley Arnott at Queen's University was leading this global experiment to test salt tolerances of a specific species of Daphnia of zooplankton. The idea being that Daphnia are a great model organism and are frequently used in laboratory settings to test a whole range of contaminants. The problem is that most of those laboratory strains have not seen the inside of a natural lake in decades. So we're talking about something that hasn't been in a lake locally, has not lived in that environment, and we might be missing, again, part of that story. So we wanted to look at this in local systems and then bring it back, and not only do it in one lake, but do it in about, I think, 45 different lakes for this project, which is exciting. So what we all did was we collected zooplankton locally, brought them back to the lab, and then cultured them and performed these salt experiments with different concentrations to see how sensitive they were at each site. So Chautauqua Lake is a participating site, and this allows us to not only explore the local effects or what are happening here, but also be part of that story to understand why that sensitivity for the same species of Daphnia might be different from one lake to the next. Is it the longer history, evolutionary pressures of having road salt constantly, like at low levels, but constantly hitting your lake? Is it something more environmental, different sorts of pressures that are important? So we'll learn about Chautauqua, but also see how we fit into the broader story. So my student Cameron went out and collected the target Daphnia species from Chautauqua Lake, brought them back to the lab, cultured them up in these clonal lines. They replicate by making clones of themselves, and then performed a lot of experiments at multiple concentrations of road salt additions to see how they survived. In these toxicity tests, we are looking for what's called the LC50, or the lethal concentration to 50% of the population. And it's just looking at what is sort of that mid-range where we start to really see population effects. So LC50 is a pretty standard metric in toxicity studies. So lots of replication, lots of controls. But what we found was that we did indeed have those two target species we were looking for, which is great. And we saw that their tolerance range on average was around about 1,000 milligrams chloride per liter. And I'll talk about what that means in just a moment. But I wanted to point out that this is, most of the time our zooplankton work just goes to genus, so it just goes to Daphnia. Cameron did some additional genetic work to make sure that we actually had the right species. So I think for maybe the first time we have actual species data for zooplankton in Chautauqua Lake confirmed genetically, which is very cool. So we have the two target species, Daphnia pulex and Daphnia pulicaria. Pulex is just a little bit more tolerant, a little less sensitive. So as that number goes up, they have a higher tolerance to that. But some big ranges too in pulicaria. What's the context for this? The EPA guideline is 230 milligrams of chloride per liter exposure, which is for a 4-day exposure once every 3 years. So that's it's a little bit weird, but it's to sort of capture that pulse. Like if high salt water comes in and they're exposed to this, they can handle it like once a year, every 3 years, they can get up to this amount of salt and still be okay. Right? There's other thresholds for shorter, more intense exposures that we could talk about more later as well. Currently in Chautauqua, our average summer chloride is at 23 milligrams per liter. So we're not anywhere close to that on average, but also that's summer data, which is not when we would expect that we would see a lot of pulses of salt coming in. So we might, again, not have the whole story. Right now we look good, right? Things are looking fine. This is a good baseline to start with, but I don't want us to get too comfortable because I think we don't have everything either, right? So we want to just make sure we're paying attention to some of those details. Additionally, it's important when we do these experiments in the laboratory that we think about how experimental conditions compare to the real world setting. We need to control, we need to have some ability to test and isolate different factors by doing these experiments. But they do miss those real world stressors that would likely make this tolerance range go down, make them more sensitive to salt exposure. In the experiments in the lab, they are fat and happy. They got nothing to worry about. They got all the food they want. Nobody's trying to eat them. Nothing to worry about. Their job is just to survive the salt. But in the real world, all those multiple stressors stack up and might make it more difficult for them to survive a salt exposure. So this is a good starting point. We'll learn more from the global experiment work that's been done. But this is, and we hope to have more conversations about road salt going forward. So those are the two main projects that my students finished up. They both did, Casey and Cameron both did excellent work this year. It's been very busy for the last 2 years in our lab. And I wanted to shift for this last portion into, as I kind of hinted at a little bit, these thoughts of timescales and thinking about what are our big gaps in our knowledge. And there's always more we want to learn as scientists, right? But when we have this opportunity to step back, we start realizing we don't have the whole story, right? As I've said a couple times, we're missing some of these key pieces. So how do we get at that? How do we go after those things that we want to know more about? And the biggest thing that's been coming up in our mind the last few years is that almost all of our data, pretty much all of our data, is gathered during the summertime, right? We have an extensive amount of data from May until late May, early June to maybe mid-September, and a little bit more maybe into October, but by then things are really winding down. We're missing the rest of those seasons, and there's good reasons for that. It's not only happening at Chautauqua Lake. That's happened in our discipline, lake sampling, for a long time, because it's easier to access, and that's when people are interacting with the system and we see those issues, right? Also, it's much more pleasant to be out on a boat when it's 70 degrees and sunny than it is to be out when it's 40 and you might fall in and freeze, right? So access is a big deal. We kind of also thought, well, there's ice on the lake, so it just must be hanging out. It's just to be dormant, right? This is a good time for us to go back to our labs and process all the stuff that we got this summer and not worry about the winter. About 15, 20 years ago, we started realizing that we were missing a lot of information, and that winter portion of what's happening in the other seasons and under the ice, there's a lot more action than we maybe expected. And so the summer conditions that we see right now are not just a product of the moment. They are a product of all of these things that lead up to that in the other seasons. So what's happening in fall, winter, spring? What's happening with ice cover and extent and quality? Ice out? When does the ice come off the lake? And what's going on under the ice? So this has really led to this question of how do other seasons affect conditions in Chautauqua, and how do we start to get more data to answer this? I liken this to, this is like, I'm trying to come up with a good analogy because I love a good analogy, but it's like you're trying to do a Lego set, because my kid loves Legos, right? And so it's like you're trying to build your Lego space rocket, right? And you know that you're building a space rocket, but you don't have more than the first couple pages of the instruction manual, and you also don't have half the pieces. And you're like, why can't I build the rocket? Why can't I figure out what's the exact trigger? Why can't I predict that thing? It's because you don't have all the pieces. And so we want to try to fill that in, right? Try to get more of that instruction manual and try to get more of those pieces at the ready so that we can see how it all fits together. And so the two ways we're doing that are to work on year-round sensor tech to put that into the lake and see if we can get that rolling. And then also a new project looking at monthly sampling. So we actually finally got to do this year. It's very exciting. In the fall of 23, in October, we started to go after this question of what's happening under the ice at Chautauqua. And yes, I use the term ice loosely because we had almost no ice this year. So it's a great year to start under ice sampling when there's no ice, but we're good. So we used our same platform, our same approach where we deployed these sensor lines again in the lake. I will point out that another reason why we don't do a lot of winter sampling is that winter is very hard on sensors. The ice likes to crush things get picked up and moved. The batteries don't like the cold. There's a lot of challenges in terms of accessing, and you can't calibrate sensors, right? It's hard to get to them under the ice, get them back, and then calibrate them. So we're limited in terms of what can actually go out. Our setup is perfect because our stuff is pretty rugged, it's pretty robust, and we can sink it down. It sinks down anyway, right? So we're below the ice, so hopefully we don't get dragged around. But sampling in the winter is not easy. But we did put out a sensor line for temperature, and then Kevin was also brave and bold and let me put his expensive oxygen sensor at the bottom of that line, which I was very worried about getting that back. We put it out in October off of, we call it our Prendergast Point location, where we've done a lot of sampling over the years, in about 12 meters of water. So I have never been so happy as to see that sensor line come back into the boat in May. You can ask Casey. I was thrilled. I was jumping around. I was like, yeah, I got it back. So I didn't even care if there was data on it at that point. I was just happy we had it and we were able to find it from under the water. It had been gone for 6 months, right? So this is very preliminary data because I said we just got it back in May and we haven't had a chance to hardly digest it yet. But I did want to show it because it's interesting. We're finding some cool stuff out already. So again, another heat map. I point out that this temperature map is a different color ramp. So my last one, it was like 25 degrees Celsius. We're talking about like 4 degrees Celsius for the red. Okay, so it's not balmy in January, it's just the color ramp takes the range. So we're talking about a much narrower range of temperature. You'll see in the first half of January, this is just the January data to show you some of the things that are going on that are interesting. The first half of January is isothermal, meaning that it's the same temperature from the surface to the bottom, which is why you have those straight up and down colors. But you'll notice that around January 16th, we get a slightly different pattern occurring, right? We start to see something that looks like stratification that we see in the summer. But you'll notice that in this case, the colder water is on top and the warmer water is on the bottom. This is a process called inverse stratification, and it happens under ice. And so it happens as a function of some weird density issues where water is densest at 4 degrees Celsius and ice floats. So the ice floats up to the top and we end up with this inverse stratification that sets up underneath. I'm going to talk a little bit more about that in just a moment, but I want to show you also our season's worth of data for temperature, or for oxygen, excuse me. So this is showing you our entire season at Prendergast Point for the oxygen sensor. You'll notice that we redeployed in October for this line, and you'll see that the oxygen increases. And it's generally high throughout the winter. But what you'll also notice is that we get these little dips in January, right? That mid to late January where we see that inverse stratification happening. We see a little dip in the oxygen. And then it resets again as lake remixes at the end of January, right? We go isothermal again. What this suggests is that there's a possibility for low oxygen to develop if we have really solid ice conditions. This was a light ice year, so things didn't have long enough for oxygen to get depleted, right? And so it got remixed again, and oxygenation was not an issue. But in some lakes, you can have what are called winter fish kills, where when the lake is capped by the ice, oxygen gets depleted and there's no oxygen for the fish. So that was interesting to observe that and see that, yes, we're well oxygenated. I don't know what this looks like under a really solid ice year. So hopefully we'll get some more ice and get to try it again. I'll also point out just briefly that in April, there's some very low oxygen values already, which is not something we would maybe expect to see until a little bit later in the season. Now, I'll caveat that by the fact that the sensor had been out in the lake for 6 months. Perhaps this isn't true or real data, right? This is what we have, but we need to get out in earlier seasons. We need to do some more April sampling to try to see if this is actually holding true. Are we really getting that low of oxygen values in the spring season that soon? If so, that's very interesting and important for us to pay attention to. So again, our first foray into under ice sampling was a success in that we got everything back and we got some cool data, so more to come on that. The last little piece I'll mention is a new project that we're working on called Thin Ice. It's funded by the National Science Foundation, and it's with our colleagues that we've worked with before on a number of other things, led by Rebecca North and others, to answer this question about how are changing ice conditions in freshwater lakes affecting algae communities. Chautauqua was invited to be part of the Gradient Lakes group, which involves monthly sampling year-round for 3 years. So it's very cool. And I have to thank Dr. Reisick because I roped her into doing this. So thank you, Allison, for doing this with me. So as you can imagine, this is a big lift. All the analytics are covered as part of the grant, but we still have to go get the samples and send those in. We'll be sampling, the plan is to sample off the bell tower at the institution. We're hoping that goes smoothly through the winter. We'll see. And as part of these samples, each month we'll collect data in the field. So we'll collect profiles for light, temperature, solid data. We'll do a Secchi reading for transparency. And then we'll collect water from 1 meter down to send to the lab for more analysis. So some of the standard things like dissolved organic carbon, nutrients, chlorophyll, total suspended solids, algae, cyanobacteria toxins as well, and then looking at the phytoplankton or algae community counts. So we started last month and we'll be going out again next week. So we're really excited to have an opportunity to participate in this project. We have lots of other things happening. I'm not going to talk about any of these, but if you have questions on some of the other things that we're rolling with in our lab, I'm happy to chat about that later. There's always more to come. Thank you especially to my current and past students. Without them, this work would not be possible. Thank you to our community partners and academic colleagues, and of course our funding groups, especially those from the Falcone and Holmberg endowments that help support our students during the summertime, which makes this, again, possible. So with that, I'll take questions. Thank you.
All right, thanks so much. If you have questions for Dr. Wigdell-Perry, you could pass them up to the front. While I'm just going to ask one that we got at the start of the presentation, and so you can think about your other, if you have other questions, and send them up, and I'll ask them now. All right, so here's the one that I got at the start, and it kind of relates to what you're just talking about. So here, we're just going to go with it. With the vast majority of phosphate being in the lake's sediment and released under anaerobic conditions, did the lack of ice cover last year result in lower dissolved phosphate levels? So you didn't talk about that exactly, but you were talking about ice, so I just wanted to give you a chance to comment on that.
Yeah, great question. So one of the reasons why we're so invested in studying temperature and stratification and oxygen is that when we have anoxic or anaerobic conditions in the deep portions of our lake, there's the potential for a shift in nutrient and shift in chemistry that allows phosphorus to be released and then become free phosphate, which is what algae and cyanobacteria love to use to grow. So that's the real reason why we're interested in like linking stratification to algae blooms. That's a great question in terms of what's happening under the ice. We don't have any data for phosphorus measured. So that's a wonderful question. And that's what we hope to get to. But that's where we don't have the data ability to get that information directly right now. So we're hoping that our winter sampling will maybe shed some light on that, and maybe some additional data for the nutrients that are coming from the Thin Ice Project will help answer that. But it's certainly a big question when we have, whether it's long duration, stable stratification, the intermittent stratification, or this under ice oxygen concentration drop, right? When that happens, there's possibilities for phosphate release. And we do see that in the bottom of the system. So the short answer is we don't know, but it's a great question. And this is part of the start of getting to that ultimately. So hopefully we'll have an update for you on that after maybe next winter. We'll see how that thin ice sampling goes, see what we get from that. So great question. But part of the real complex story that we're telling and linking all these conditions back to the algae blooms that we care about right now.
Awesome. Thanks for that. Okay, got another question here. If I'm reading this wrong, please let me know. Have you considered studying temperature stratification versus plant density and the implication for HAB conditions? So I guess just trying to figure out if those things play together and if you've considered studying it.
We always get great questions here. I love doing talks down here. So we have not studied that directly. The plants are typically living in the shallower portions of the lake, right, along the littoral zone, the shoreline zone. And so at Chautauqua Lake especially, stratification in the summer months, that layer, right, is usually hitting around maybe as shallow as 6 meters depth, but typically hanging out more around 10 meters depth. So most of the plants in the lake are really not growing past 3.5 meters, maybe 4. So in terms of like where the plants are situated and where stratification boundary happens, it's not directly impacting that. Now, as I said, that boundary can flex, and there's certainly possibilities for interactions at the end of the lake as we sort of crack the whip, right, into the sediments. The most direct connection I would see is that nutrient release from the sediments due to anoxic conditions could help fuel the plants, right? Plants and algae and cyanobacteria are all looking for those same nutrients to grow. So in terms of direct connection, the stratification itself is probably not directly affecting it, but likely through any sort of nutrient release that could fuel that. The link between algae and plants in this lake, it's not been thoroughly studied to understand, is it a positive relationship? Is it mutualistic? Do they both benefit? Is it antagonistic, where they are in competition for the same nutrients? And likely that's not a one-size-fits-all category, right? Probably some species are in competition with each other, and probably some are mutually benefiting from them being there. So it's a great question that has more to that story, but in general, when we have high nutrients, we're going to have high productivity by both plants and algae and cyanobacteria. So we should be paying attention to where those nutrients are traveling, and that's part of understanding that internal nutrient budget, thinking about where those nutrients are coming from in the lake, where are they coming from outside of the lake, and how does that move, right? What happens to water when it comes into the system, which I think we'll hear more about maybe later today, how water moves when it gets into the system, tracking some of that chemistry, and then physically watching where it goes, right? Does it stay in the shallows? Does it move down to the bottom of the system? We don't know. We're hoping to get at some of those things this year with some short-term sampling, but it's a great question. So there's lots more to the story there to understand nutrient pathways and cycles and get a better sense of that budget. I think that'll help us see what the effects are on multiple biological communities.
Awesome. Okay, I don't think we have any other questions right now. So thanks so much.